All right, I think we're at 1:30 now, so I think we'll get started. And maybe a few more people will trickle in as I give the introduction, but welcome everyone to today's workshop on reference electrodes hosted by WatECS and Dr. Drew Higgins. My name is Jillian. I am the WatECS president, and our group, if you're not familiar with us, we plan workshops and events to connect researchers and graduate students in the field of electrochemistry. If you are familiar with us and have been to some of our previous events, we're actually looking for some new executive team members. So I'll post a link in the chat box for anyone who is interested in joining our team or applying. But today we're very excited to be hosting the last workshop in our electrochemistry techniques series, hosted today with Dr. Drew Higgins from McMaster University on reference electrodes, something very fundamental that all of us use in our electrochemistry research on a daily basis. So I wanted to give a bit of an introduction to Dr. Drew Higgins, who is an assistant professor in the Department of Chemical Engineering at McMaster University. His research focuses on the synthesis and characterization of nanomaterial catalysts for electrochemical CO2 reduction and low temperature fuel cells. So this is really exciting. We're really excited to have you here with us today. As a bit of a housekeeping note before we get started, if everyone wants to stay muted throughout, feel free to post your questions in the chat box and we can answer them either throughout or at the end of the workshop. Also, if you're attending from chemical engineering at University of Waterloo, this will count for your seminar attendance. So we'll post a code in the chat box 15 minutes before the end of the seminar. And I think that's everything on my side. So thank you all for attending and thank you, Dr. Higgins. I'll let you take it away.
Awesome. Thank you, Jillian, and thank you all for being here today, and especially to ECS Waterloo for the invitation. I should mention I spent 10 years in Waterloo as an undergraduate student and as a graduate student. So it's a city and a university that's very near and dear to my heart. And to be honest, the reason that I first got started on electrochemistry is because I took ChemEng 331, the electrochemical engineering course taught by Michael Fowler at the time in the chemical engineering department at Waterloo. And I really thought it was exciting. You know, electrochemistry provides an opportunity for chemical engineers to contribute to sustainable energy technologies, you know, the things that are going to power our future. And, you know, I think electrochemistry courses should be a key part of every chemical engineering undergraduate curriculum. And I was surprised to find out that it actually isn't. So Waterloo has it. That's great. That inspired me back in the day to pursue this direction. Here I am, you know, 10 years later, still doing it. And so I'm actually going to be developing an undergraduate course that's going to be offered at McMaster to hopefully inspire the young generation to go in this direction that I think is so important for the sustainability of our future. As Jillian mentioned, my name's Drew Higgins. Please feel free to call me Drew. And today I'm going to talk a little bit about reference electrodes, particularly the selection, the validation, and the use of reference electrodes for electrochemistry experiments. And more particularly, I'm going to talk about experiments that involve the operando or the in situ characterization of electrochemical processes. Now, I wasn't exactly sure the best way to put this together. I didn't necessarily have like a workshop on reference electrodes prepared. So I kind of made like a hodgepodge of slides. I had prepared some new slides to put together a bit of a workshop flow. I hope it works. But what I'm assuming here is that most of you have at least a basic level understanding of the concepts of electrochemistry. But I tried to not leave too many gaps, nor dive into the weeds too much to lose people in the process. And I hope I'm successful at this, but please feel free to speak up at any time or post something in the chat. I've got the chat open and I'll monitor it because I'd hate to lose someone on slide 2, you know, of a 50 slide deck. So please feel free, if there's something you don't understand, we can loop back to it. So just in terms of the motivation for why one might do electrochemistry related research, you know, since the industrial revolution, we've seen significant increases in our global temperatures that leads to significant consequences in terms of climate change, weather patterns, food shortages, etc. And the reason this is happening is if we take a look at our energy portfolio, at any given time, the world is consuming about 18 terawatts of energy. And 85% of that is coming from fossil fuels with associated CO2 emissions. If we stay on this trajectory, we're going to see even more devastating consequences of our CO2 emissions, when really I look at it as the job of scientists and engineers to develop new processes and new technologies to get us on this more safe trajectory where we can really limit our emissions and thereby limit our global temperature increases and all the devastating consequences of that. So this is the vision that I share with many of my colleagues of what a future sustainable energy technology landscape may look like. So ideally, we want to rely on renewable sources of electricity, things like wind, things like solar, things like hydro. And then we want to be able to use that electricity to create the fuels, chemicals, and fertilizers that society depends upon. But we want to be able to do those from precursor molecules that are readily abundant to us. So things like nitrogen, things like CO2, things like water. If we could combine these abundant, benign molecules with electricity to create the fuels, chemicals, and fertilizers that society depends upon, I think we'd be able to achieve a fairly sustainable energy landscape. So the key thing, and what the research my group focuses on, the research many of you focus on, is all of these processes rely on electrochemical conversion technologies, and more specifically, catalysts or active materials that drive those electrochemical technologies. So for example, the conversion of CO2 into fuels and chemicals, low temperature fuel cells for transportation applications, for example, or for example, battery development for things like electric vehicles or for grid scale energy storage that will allow us to increase the penetration of renewables into our energy landscape. So this is just a quick overview of the research that my team does, broken into the different fields that we work in. We have some projects on supercapacitors with the automotive company. They're interested in incorporating those into their electric vehicles. We work on electrochemical conversion of CO2 into fuels and chemicals. We have a project on rechargeable zinc ion intercalation batteries that are being targeted for grid scale energy storage. We have some projects looking at the electrosynthesis of chemicals. And then a large part of our research, and I'm going to touch on this a little bit today, is on the in situ or operando characterization of electrochemical catalyst materials or electroactive materials in the case of a battery, for example. So all of our research focuses on developing new nanomaterials, understanding them, and implementing them into a prototype technology. And really, so how do we do that? So the way we do that is we have a feedback loop that we follow in our lab. So, you know, first off, we have a challenge we want to address, whether it's the performance of a CO2 conversion device or a fuel cell, or the energy storage capacity of a battery. And we go to the literature and we use our chemical intuition to come up with new nanomaterial designs that will hopefully address the challenges that are plaguing those fields. We synthesize these materials in the lab, and then we test them for their activity, their selectivity, their stability, their energy storage capacity, and we correlate these performance evaluation results from our performance evaluations with the results of our in situ or operando characterization. So the characterization of these materials while they're operating, to understand these performance descriptors, or essentially what properties, what structures, what traits of these material give them good performance. Once we have that fundamental scientific insight, we can feed that back into the design of new materials with hopefully improved properties, improved performance. Ultimately, we try to arrive at something that's better than anything that's ever been seen before, and we integrate it into an actual device for prototype demonstration. Now, a key part of that is the linking the in situ or operando characterization with the performance evaluation of these materials. And ideally, we can also couple these experimental results with computational theory, such as DFT or Monte Carlo simulations, to really understand what traits of active materials make them good, guide new designs of active materials with better performance, or be able to even predict new compositions or new structures that will give us unprecedented performance capabilities to really allow us to develop and commercialize these important sustainable energy technologies. Now, the key thing here is that when we are linking these results of characterization and performance evaluation, it is so important that the results of our characterization are accurate, that these characterization techniques are telling us the actual properties and structure of these materials, ideally under reaction conditions, because knowing what their properties are under reaction conditions is the ultimate descriptor of what makes either a good material or a bad material. And so this all feeds into our design. And if we have information that's not correct feeding into our design, that can cause a lot of problems. So we want to make sure that these characterization results are correct and are guiding our scientific efforts in a rational way. So that leads me to the next point, where first, I think we can just talk about a little bit of definition. So a lot of times you hear the terms being thrown around in situ characterization or operando characterization. They're similar, but there are some subtle differences. When we say in situ characterization, that means studying a material under relevant reaction conditions. If we want to take that one step further, we can talk about operando characterization, which is also studying a material under relevant reaction conditions, but it's in a more practical sense where we're simultaneously measuring the activity and the selectivity or the durability of these materials. We're getting some sort of performance metrics out of it, coupled with the characterization under reaction conditions. And now I just kind of want to highlight why operando or in situ characterization might be important. And I always like to use the detective analogy. So a lot of times when people are trying to understand the performance of materials, what they'll do is they'll synthesize the materials and they'll characterize those materials. And that will be their understanding of what the structure and properties are of their materials. Or what they'll do is they'll synthesize materials, test those materials out, and then characterize them after those tests to understand what the structure and performance is after those tests. But commonly, the properties of the materials during testing are not the same as the properties of materials either before or after those tests. So I like to use the detective analogy where you can take a look at a crime scene. You can take a look at the crime scene before a crime is committed. You can take a look at a crime scene after that crime is committed. And at the very best, all you can do is speculate about what happened while that crime is being while that crime is being committed. At the very best, you can guess. But if you can do in situ or operando characterization, you're essentially shining a lens on what is happening during that reaction. Or in the case of a crime, you can see exactly what happened when that crime is committed, and you have all of that information, and you have that information with a high degree of certainty. And so if you have that correct information, that means when you're feeding that into your scientific understanding, you're feeding that back into your research efforts, you are operating with a lot more accurate information to guide your future studies. So hopefully this demonstrates to you why, at least I feel, that in situ or operando characterization can be so powerful to guiding our research efforts. Really, what these techniques can do is they can give us insight into how electrodes perform, how catalysts perform, how the different species in a three-dimensional electrode come together. And there's all sorts of processes happening on varying time and varying length scales that having this operando characterization will let us disentangle, let us understand all of these processes going on in these materials, and that once again can guide future material designs, advanced material designs with better performance. So there are a lot of different characterization techniques that have been used, generally broken into optical, scanning probe, electron, or X-ray techniques. And breaking it down even further, there are many different characterization techniques that can be employed to characterize different regions of an active material. For example, we can characterize the actual catalyst or the active material in the case of batteries, or we can characterize that catalyst electrolyte interface, or we can characterize the electrolyte. Different characterization techniques are relevant to different regions and can give us different information about all of these processes going on. And so I should mention the framework for this talk was brought together by a paper that Wajdi Alnoush, a PhD student in my group, published late last year. So I encourage you to take a look at that if you want some more details or you want some more information. But a lot of these figures were plucked right out of that paper. So what are in situ or operando experiments? So essentially they are simple electrochemical measurements, the type that anyone runs in an experimental electrochemistry lab, but the reactor has been designed to accommodate characterization probes, whether those are electrons coming in and going out, X-rays coming in, going out, light, what have you. And there's some key features that need to be designed to make these in situ or operando experiments successful. The first is the geometry of your electrochemical reactor. The second is how the electrolyte is handled. Is it flown in? Is it mixed in? As well as how the reactants and products are fed to your active materials or removed from your active materials. And then also, and this is important, of course, in the case of electrochemistry, the control of your electrode potential or the control of your electrode current. And I'm going to be talking about this in great detail because the reference electrode plays such an important role on this third point that I put on the slide. We need to be able to accurately control the potential of our electrode and measure the currents that are going on in our electrochemical cell. So there are different types of electrochemical cells used for experimental electrochemistry, and these may, you may have used some of these or several of these in your research. The first is the traditional 3-electrode cell, where you have a counter electrode, you have a working electrode, and you have a reference electrode. Then on the flip side, we have a more of like the practical electrochemical device, something like a battery, where you have 2 electrodes. So a working electrode, we can, for example, call this the anode, and the counter electrode, or we can call it the cathode. But really, there's 2 electrodes going on. And I'd just like you to take a quick look at this picture and take a look and see what the key difference is between this 3-electrode cell and the 2-electrode cell. Obviously, it's in the name. One has more electrodes, but the key difference is this reference electrode here that's used in this 3-electrode cell. And the purpose of the reference electrode, this reference electrode is important because what it does is it allows us to know exactly what the electrode potential is at the working electrode. We know exactly what the electrode potential is at the working electrode when we have a reliable reference electrode. So this is very handy when we're doing experimental or analytical electrochemistry, because the potential of that working electrode dictates all of the thermodynamics and dictates all of the kinetics of whatever it is that we're probing. So this reference electrode is very important, and it's very important that it's operating properly. Now, just quickly before I go any further, one common misconception or one common error that I see many students or even many experienced electrochemistries use is the difference in nomenclature between electrode potential and voltage sometimes gets mixed up. So I just want to highlight quickly that if you are measuring the electrode potential difference between a working electrode and a reference electrode, that difference in potential is referred to as an electrode potential. If, on the other hand, you have a 2-electrode system and simply you're measuring the difference in voltage between, the difference in electrode potential between a cathode and an anode, this is referred to as a voltage. Or you can call it a cell voltage. There's two there's a fundamental difference between these two terms. And I encourage all of you to make sure that when you're describing your electrochemical experiments or your electrochemical systems, that you're using the correct nomenclature. Otherwise, someone could find it very confusing if you're mixing up electrode potential and cell voltage, for example. Now, just to reiterate that, the electrode potential is the electrical potential difference between an electrode and a reference electrode. And in this case, between your working electrode and a reference electrode, there is no current flowing between the electrodes. And the reason we use a reference electrode is because the absolute potential of a working electrode cannot readily be measured on its own. We always need to measure it versus an arbitrary 0 point. And this arbitrary 0 point is generally the electrode potential of a well-known redox pair or a reference electrode potential. On the other hand, the voltage is the potential difference between an anode and a cathode electrode in an electrochemical cell. And in this case, there would be current flow between the two electrodes. So just a quick lesson in nomenclature and something that I encourage all of you to make sure you understand and are using the correct terminology in your research. So when we're talking about a 3-electrode system, just dive into the different components quickly here. We've got our working electrode where we've got our active material or our catalyst. It's important to consider what the surface area of those materials is. So the geometric surface area or the 2-dimensional footprint of that electrode, as well as the 3-dimensional surface area, often referred to as the electrochemical active surface area. We also want to know the geometry, where it is in the cell, because there's various potential gradients that may form in that cell, as well as the surface condition. You know, is the surface of your electrode an oxide? Is it poisoned by something? Is it metallic? All of those can make a difference on your experiments. Then we've got the reference electrode, which we're going to talk about more today. You know, and you may have the question, should I run my experiments with or without a reference electrode? And if I am going to use a reference electrode, which one? And if I do choose a reference electrode, how can I calibrate it? How can I validate it? How can I use it so that my experiments are done as reliably as possible? And that's something we're going to talk about a lot today. Then you've got your counter electrode. There's a material selection that goes into that. In some instances, you might want to use something like a platinum wire. In other instances, you might want to use a carbon rod, or maybe there's another option that suits your experimental purposes. And it's always good to know what reaction or process is occurring on your counter electrode, because that can have implications on whatever the experiments are that you're doing. So just make sure that's something you consider. And obviously, you want to make sure that whatever counter electrode you're using is not going to contaminate whatever the process is that you're probing with your experiments. And then last but not least, you've got your electrolyte that sometimes has the reactant solubilized within it. Sometimes the reactant is fed separately to your electrode. But in that electrolyte, you're going to have a solvent, you're going to have redox species, you're going to have reactants, you're going to have products. So all of these things are important things to consider when you're running your experimental measurements. Here's just a picture of a few of the 3-electrode systems that I've seen before. If anyone does oxygen reduction studies, for example, you'll see this RDE cell. This 3-electrode cell shown here is what we use in our lab for electrochemical CO2 reduction experiments. There's really kind of tons of designs. Some people custom design electrochemical cells, 3-electrochemical cells for their purposes, but they come in pretty much all shapes and flavors. With that said, reference electrodes come in all shapes and flavors. And the key thing that a reference electrode does is it provides a defined potential versus which the electrical potential of the working electrode can be reliably measured and controlled. And this reliably word is very important here. We want to make sure that we can reliably control and measure what is happening at our working electrode. And just to reiterate that point or place a underline under it, a stable an accurate and reliable reference electrode is a prerequisite for analytical and experimental electrochemistry, including operando characterization. It is so important for making sure that our experiments are done accurately and done reliably. Just quickly, when I do say a defined potential versus which the potential of a working electrode can be measured, that defined potential is essentially the equilibrium potential or the reversible potential of the redox couple in our reference electrode. So it's really important that, as I've talked about, that we can calibrate our reference electrodes so we know what their electrochemical potential is, and that we validate that that electrode potential of the reference electrode is not going to drift with time. Because if our reference electrode drifts with time, all of our experiments are going to become inaccurate as a function of time. So it's really important to calibrate it and make sure that that value stays constant. We also want to have a redox pair that is reversible. So for example, an RHE has hydrogen proton redox pair, very, very reversible reaction. Or, silver chloride has a very reversible redox pair between silver chloride and silver and chlorine ions. The reversibility is very important when you're thinking about a reference electrode. You also want a reference electrode that's non-polarizable. And this means that even if small amounts of current go through the reference electrode, which generally they shouldn't, but it can happen for one reason or another. But you want, if a little bit of current goes into your reference electrode, you don't want the potential of that reference electrode to shift dramatically. These two things are generally coupled, because if a redox pair is reversible, that means it doesn't have high over potential going either in the oxidizing or the reducing direction. Then you also want a reference electrode that is not going to contaminate your experiment. It's not going to contaminate your electrolyte. It's not going to contaminate your working electrode. It's not going to interfere with the targeted electrochemistry of interest. So say, for example, you're running you're using a silver chloride reference electrode, and some of the chlorine ions leach out of your reference electrode into your electrolyte. For something like platinum, chlorine ions can interfere very strongly with some of the electrochemistry. So you just want to make sure that whatever reference electrode you're using is not going to contaminate your experiments. Now, in case you have not gotten sick of me saying how important a good reference electrode is, I'll just provide a real example of why a reliable reference electrode is important. Let's take, for example, the case of oxygen reduction on carbon-supported platinum. So platinum is a very good oxygen reduction catalyst. And say we have a reference electrode that is 100 millivolts off from where we think it is. What can happen then with a 100 millivolt difference between the measured potential and the real potential of our reference electrode? We can have almost a 2 orders of magnitude difference in the currents that one would expect. So at about 0.95 volts versus RHE, platinum would generally have a mass activity of about 10, maybe 20 milliamps per milligram. If we have a electrode potential of 0.85 volts versus the RHE, we're going to be up on the order of 200 milliamps per milligram of platinum. So huge differences that could be convoluted if you have a reference electrode that's about 100 millivolts off, or even 50 millivolts off. Imagine the worst case scenario if you have a reference electrode that is several hundred millivolts off. This can provide a lot of issues with your results and with your interpretation of those results. And so it's best to take these into account before you run your experiments and make sure that you're carefully selecting, using, and validating, your reference electrodes. And so there are many different types of reference electrodes, and I'm going to talk about 3 different categories today. I'm going to talk about conventional reference electrodes. I've already talked about silver chloride, for example, or the reversible hydrogen electrode. I'm going to talk a little bit about miniaturized reference electrodes. So some of you may have used these in some cases where your experimental electrochemistry is a little bit geometrically confined. And then I'm going to be talking about quasi-reference electrodes, which are commonly used if you're doing non-aqueous electrochemistry, or in our case, if we're doing electrochemistry on microchips, where we are significantly spatially constrained to the size of reference electrodes we can use. So first, I'll talk about conventional reference electrodes. These are the ones that are commercially available. They have well-defined redox chemistries. So for example, silver chloride, we've got our mercury sulfate, we've got our saturated calomel electrodes. There's lots of different well-defined reference electrode chemistries that you can just buy, for $100, $200 from VWR or whatever supplier. Lots of them available. They're stable, they're easy to use, and like I said, they're relatively inexpensive. But with that said, even though they are relatively stable, I just want to highlight they should still always be calibrated regularly, because over time they can drift by tens of millivolts, sometimes as much as 100 millivolts. So now you may be asking, okay, well, how do I calibrate my reference electrode? And when should I do it? To answer the when should I do it, I would always say that calibration, in an ideal world, should be done before and after every experiment. Once you do that a couple times and you prove to yourself that your reference electrode is stable over the time period of a couple of hours or a couple of days, okay, maybe you can decrease the frequency that you calibrate it. And, but at the very least, I would still recommend you calibrate it every couple of weeks or maybe once a month. And to answer the question of how one can do it, there's a few different ways. I'm going to talk about 2 on this slide. The first is by calibrating your reference electrode versus a reversible hydrogen electrode. And a reversible hydrogen electrode is essentially a platinum wire immersed in the electrolyte that you're using with hydrogen bubbled over it at 1 atmosphere. It's very easy to make a reversible hydrogen electrode. I encourage all of you, any of you electrochemists, I encourage you to learn how to make an RHE, be comfortable with the concept of an RHE and how to actually implement it into the lab, because it's a very handy experimental tool to have in your toolbox when you're doing electrochemical measurements. And so you make this homemade RHE. You stick your commercial reference electrode in that same electrolyte solution with the RHE that you've made, and then you just simply measure the electrochemical potential difference between the 2. And then you've got a calibration value to convert all of your potential measurements versus your reference electrode to the RHE scale, which is kind of the universally used scale in many fields of electrochemistry, especially in electrocatalysis, where my team works largely. Another way to do it is to have what one might call a master reference electrode. So it's a commercial reference electrode that you store away in its proper storage solution. You give it all the tender love and care. You take good care of it. You protect it with your life, so that you know that this thing has been treated very well, and it's going to be very stable. It's not going to drift over time. And you can always measure your other reference electrodes, the ones you're beating up on a daily basis, you're using all the time. You can measure the potential of the 2 versus each other to calibrate the reference electrode you use all the time to this master reference electrode that in principle should not have drifted from its default value. I prefer option 1, because you would never know if your master reference electrode drifts, for example, whereas the reversible hydrogen electrode is very well-defined electrochemistry, very easy to make, and it's going to be exactly at 0 volts versus the RHE if you build it and operate it properly. The one quick tip I would give is, if you do make your own RHE, is always make sure to butane torch your platinum wire before you use it. Because that just gets off any junk, any organics, any species that may have built up on the surface of that platinum wire and could impact your measurement. And then I'll give you a second tip, and this one I learned the hard way. Make sure you let that platinum wire cool down after you butane torch it before you stick it into a beaker where you're bubbling hydrogen. And you can imagine what mistake I might have made in the past. So now let's just give a practical example of when a conventional reference electrode could be used for in situ characterization of materials. And so during my postdoctoral research, we were developing these copper silver thin films, and we found using X-ray diffraction that it was a multi-phase system consisting of copper rich regions with silver atoms sprinkled within it, and then silver rich regions that had a few copper atoms within them. So we call these our metastable phase segregated materials. That's not really the point, though. The point is that when we tested them for their electrochemical oxygen reduction activity in KOH, we found that the copper silver mixed metal thin films significantly outperformed pure silver. And pure silver is one of the best known oxygen reduction catalysts in alkaline electrolyte. And so essentially, if you've never seen one of these oxygen reduction polarization curves, just simply anything that is to the right and down on this axis means a better catalyst. And so from just looking at this plot, you can see that copper silver was our best catalyst, silver was our second best, and copper was our third best. And it was interesting to us that the mixture of copper and silver performed much better than silver, a well-known ORR catalyst, and much better than copper. So these two things were not operating like the sum of their parts. They were coming together and providing a significant boost in activity, and we weren't sure why. We also saw this hysteresis in our polarization chart, where in the upward sweep direction, the activity was better than in the negative potential sweep direction when we were doing our cyclic voltammetry. And so we want to know two things. We want to know, why is copper silver better than pure silver or better than pure copper? We also want to know why this hysteresis was occurring in these CVs. So we packed these materials up, and we designed a electrochemical cell that would allow us to get X-rays in to the copper silver thin films and get fluorescence out from the copper silver thin films using a technique called X-ray absorption spectroscopy. And so this is a synchrotron based technique that we were doing at SSRL down at the SLAC National Laboratory in California. And we designed a reactor where we needed to have a reference electrode in it. And so we designed a reactor that could accommodate a conventional silver chloride reference electrode. We set these materials up in front of our synchrotron X-ray beam, and we collected X-ray absorption spectroscopy at the copper K edge. And now I don't want to dive into the weeds of this particular technique, but really we wanted to characterize what the oxidation state and what the structure was of these materials under operating conditions or under electrochemical potentials, where that reference electrode was really important for making sure that we had reliable electrochemical potentials, because we were comparing the results from one experiment to another. On one hand, pure copper or pure silver to the copper silver mixture, and we want to make sure it was apples to apples. When we were looking at 0.75 volts versus the RHE for one catalyst, we want to make sure we are comparing those results at the exact same potential for another catalyst. So that's where the accurate, the reliable reference electrode becomes very, very important. So I know this all looks like a bunch of squiggles. I'm not going to dive into the weeds of this, but essentially what we found is that when we compared our copper silver catalyst to pure copper, we found that the copper atoms in our copper silver thin films were in the metallic state when they were in copper and silver, whereas when it was pure copper, they were in their oxidized state, and copper oxide was not able to do the oxygen reduction. So what we did is we essentially identified that it was the metallic copper atoms in silver copper, where the presence of silver allowed those copper atoms to maintain their metallic state, that gave us that boost in activity. It also provided the hysteresis that we observed, because the hysteresis was caused by the oxidation and the reduction of those copper atoms. And we did a bunch of density functional theory. We collaborated with one of our colleagues to corroborate these understandings. And I see the question in the chat, which composition of copper silver was better performing? We found that copper 70 silver 30 was our best performing catalyst thin film, but we found that at most compositions, the copper and silver perform better than just the silver alone. But 70-30 was our best one. So I talked a little bit about conventional reference electrodes. They're nice, they're convenient, they're stable, they're inexpensive, but they are, if you look at them, they're about 1 to 2 centimeters in diameter, which is not that big, but it is too big for some applications. Which brings us to the next category of reference electrodes that I want to talk about, and that's miniaturized reference electrodes. And these miniaturized reference electrodes are on the order of a few millimeters in diameter. And going down to a few millimeters in diameter can help us overcome some geometrical constraints. And so these miniaturized reference electrodes can come in 2 forms. One is by using a Lugin capillary, which I look at as essentially as an extension cable for a reference electrode. It's just a small plastic tube with a glass frit on the end that you can fill with a reference electrode fill solution and essentially extend the sensing area of your reference electrode into a small electrochemical cell, for example. Or there are commercially available miniaturized conventional reference electrodes. For example, miniaturized silver chloride reference electrodes that have 1 to 2 millimeters diameter. Now, I'm just going to give a quick public service announcement. And, if you've had different experiences, I'd love to hear about them. But I should note that these miniaturized conventional reference electrodes, these 1 to 2 millimeter silver chloride reference electrodes, can be very unstable and very unreliable. I found them drifting by as much as 300 or 400 millivolts from day to day. And so if you treat them well and all the stars align, they can be very reliable, they can be very convenient. But if you are going to use those, be careful and make sure you are calibrating them in a regular at regular intervals. So we have some questions about the copper versus the copper silver. Maybe if we could loop back to those at the end, and I actually don't have anything set up after the seminar, so I can stick around a little bit longer and chat about that a little bit too. I just worry that it might detract from the main point of the talk. So Shajil, please stick around at the end. So talked a little bit about miniaturized reference electrodes, and so maybe I can just show an example of where these have been used for operando or in situ characterization techniques. And so we were developing thin film catalysts to try and gain fundamental insight into the electrochemical CO2 reduction reaction. And when you're using a thin film, you've got 50, 100 nanometers of metal deposited on a substrate. But we were interested in these materials as catalysts. And we know that catalysis is a surface-based process. So we wanted to be able to characterize the surface of these thin film materials. And one way to do that is to do measurements in what's called grazing incidence configurations. So essentially, you illuminate your catalyst thin film with X-rays, but you illuminate it at a very glancing angle. So it just glances off the very surface of your catalyst, and that can give you near surface information about the near surface region of your catalyst. This is in contrast to conventional measurements where you just blast your thin film with X-rays. It can be perpendicular at any other angle, and you're essentially illuminating the bulk at that time, and the spectra that you're going to get is going to be a bulk averaged spectra of your materials. So we needed to design a reactor that we could get X-rays in at very glancing, very grazing incident angles. For these measurements, we're doing combined X-ray absorption spectroscopy using a fluorescence detector, and we are also doing X-ray diffraction using an area detector. So we had one detector off to the left of our electrochemical cell, and an X-ray diffraction detector that was kind of looping around the top of our electrochemical cell. And so we had some significant geometrical constraints when we were designing, when we were using these cells. And so this is essentially what the 3D printed electrochemical cell looked like. This was designed by a beamline scientist at SSRL named Ryan Davis. But because we were so geometrically constrained, we only had a very small area where the reference electrode could be inserted into this electrochemical cell. And so you can see here, this little blue nut here is where a reference electrode went in. You can kind of see it on this diagram here. We didn't have much room to work with, so we used a 2 millimeter silver chloride reference electrode for these experiments. And this is where some of those headaches that I mentioned came from, where we found that this reference electrode wasn't as reliable as we would like it to be. But really, because of our space restrictions, we had no better options. And so we were like good little scientists. We made sure we calibrated, and we did a lot of sanity checks on those materials to make sure that our measurements were done in a way that the electrochemical potentials we were measuring and reporting were reliable. So we flowed our electrolyte in with our CO2 reactant in it. We applied electrochemical potentials being controlled by the reference electrode, and we did X-ray absorption spectra measurements, and we did X-ray diffraction measurements. And once again, I don't want to dive into the weeds of these techniques, but essentially in the literature, or if we want to go based on as prepared catalyst or post-testing catalyst characterization, if we were trying to understand the CO2 reduction process on copper, we would think that CO2 reduction occurred on metallic copper covered by a thin layer of copper oxide. But when you actually do the in situ or the operando measurements, what we found is that there was no copper oxide present at all, or no copper oxide present at all. It was all metallic copper. So what that told us is that the CO2 reduction was occurring on metallic copper. And this was the only way we could get conclusive evidence of what the surface of those materials looked like under reaction conditions, was by doing it via this in situ or operando characterization enabled by one of these miniaturized reference electrodes that I talked about. So that's all great. You know, there are some challenges using these miniaturized reference electrodes, but they can be overcome if they're used properly. But even when we're talking about diameters of 1 to 2 millimeters, that can still be too big for some purposes. So that brings us to what these purposes may be. And these may be experiments where we require microchip reactors for certain in situ characterization measurements. And particularly the ones that we work on are transmission-based measurements. So for example, transmission electron microscopy. So the reason why we need these microchip reactors is because if we have too much electrolyte, we're never going to be able to get enough X-rays in or electrons in and through the sample to our detectors. So we need to have these small reactors where we have only a couple hundred nanometers of electrolyte in there. And the only way to reliably do that is with these microchip reactors. Now, because these microchip reactors are so tiny, there's no way we can get a 1 to 2 millimeter silver chloride reference electrode in there, for example. So we use what is called a quasi reference electrode. And they allow us to overcome some of these spatial restraints. So I mentioned that they're generally used for transmission-based measurements. Here are 2 areas of research that my group is actively working, where we are commonly using quasi-reference electrodes in these microchip reactors. We're doing a lot of in situ electrochemical transmission electron microscopy. You can see Ahmed Abdallah, a PhD student in my group there, putting his microchip reactor into a TEM. Or on the right, we're doing a technique called in situ scanning transmission X-ray microscopy. So that's a mouthful, but essentially what we're doing is we're able to do microscopy on materials where each pixel in that microscopic image is actually a spectra that can give us chemical information about our materials. And we're collaborating very closely with Professor Adam Hitchcock, who's here at McMaster University in the Department of Chemistry and Chemical Biology, really a world leader in this STIXM technique, and Chanyang Zhang, a PhD student in my group, who was co-supervised by myself and Professor Adam Hitchcock. So just diving into the details of what these measurements may look like, what these microchip reactors may look like. And it's essentially 2 chips on top of each other, sealed together, that we can flow electrolyte into. Within the chips, we have these silicon nitride viewing windows, and silicon nitride is essentially X-ray or electron transparent. So we can just fire electrons or X-rays through that silicon nitride window, illuminate our working electrode where our catalysts or our active materials are developed. And if we zoom into what these microchip reactors look like, we can see it's really just a conventional 3-electrode configuration. We've got our working electrode here in the middle with our active materials deposited on it. We've got a platinum counter electrode that kind of surrounds our working electrode. So it's this C-shaped electrode. And then we have our small reference electrode, which in this case is just a platinum metal quasi reference electrode. And so we have a question, quasi, why is it called quasi? So the term quasi or pseudo reference electrode are sometimes used interchangeably, and essentially quasi means Latin for approximately. So quasi means that these reference electrodes, they're not like a well-defined reference electrode in the way that silver chloride is, but it essentially suits the same purpose. So that's why it's called a quasi reference electrode. And then just looking from the side angle, so we've got our working electrode here, where we have our active material illuminated in the top. And then in the case of transmission electron microscopy, our electron beam comes in, goes through our catalyst materials or our electrode materials, and then comes out where it goes to the detector. I apologize for my terrible artistic capabilities, but essentially this illustrates why we need this very thin electrolyte layer. Because if it's too thick, these electrons are never going to be able to get in, and they're never going to be able to get out, and we're never going to be able to characterize our materials. So this is just why we need these microchip reactors. So in terms of quasi-reference electrodes, so they're typically noble metals such as platinum, such as silver, such as gold. Reason being that noble metals are, as they're named, noble. They're not going to form metal oxides on the surface that would plug them up, that would change their surface chemistry too much. The nice thing about them is they don't have any liquid junctions where you can have IR drops or you need to deal with reference electrode fill solutions. They're also generally very high purity. There's a lot of techniques in the microfabrication community to prepare metal wires or evaporated thin films of these metals, so they can be very high purity. You don't need to worry about contamination. And because of all the work that's been done in the microfab community, they can be manufactured in many different sizes, many different geometries, even down to these very, very, very small sizes that we need for these experiments. So a few things to note is that their reference potential is determined by the redox pair that these metals establish with whatever the species are present in your electrolyte. And typically this is the metal interacting with the water molecules in your aqueous electrolyte to form either a metal oxide or a metal hydroxide. So it's this metal oxide or metal hydroxide redox pair that defines the electrode potential of your reference electrode. So it's not as well defined as a conventional reference electrode, but with enough care and control over it, you can have an idea of what the redox processes are occurring on your reference electrode and that are dictating the potential of that reference electrode. But it's important to consider that these are susceptible to changes within the local environment. So say, for example, if your working electrode is generating a bunch of hydrogen and your reference electrode is a quasi reference electrode of platinum wire, it's going to interact with that hydrogen, and it's going to form a mixed potential where the hydrogen proton redox couple, the platinum hydroxide redox couple are kind of have this interplay, and you're going to have a weighted average of those two potentials as your reference electrode potential. So you always want to take care and be sure of what species are being formed near that reference electrode, because that can impact the potential of your reference electrode. So that's why I say care must be taken when you're selecting, validating, and utilizing these quasi-reference electrodes. So I'm running a bit on time, so I'll speed it up a little bit. Essentially, we want to ensure the compatibility of your quasi-reference electrode with your electrolyte solution and species. For example, if you're going to use a gold-based quasi-reference electrode and you've got a bunch of sulfur-containing species in your electrolyte, those sulfur species are going to poison your gold electrode and pretty much ruin your pseudo-reference electrode, your quasi-reference electrode that's based on gold. Always need to consider the cost, ease of fabrication, and the reproducibility of these quasi-reference electrodes. And then just the size and the geometry of the electrochemical cell and experimental conditions that you're going to be employing. So how do you validate these quasi-reference electrodes? And so just like any reference electrode, regular calibration is so important. We want to make sure that these quasi-reference electrodes have a well-defined potential and that it is stable over the course of measurement. And that's one thing that's missing in a lot of scientific papers I read that use quasi-reference electrodes is they don't provide that much in the way of details of how they calibrate, how they validate, and how they use their quasi-reference electrodes. So if you are using quasi-reference electrodes and you're writing a paper, make sure you put in all the details, because I think that's really important for other researchers to understand the implications of using a quasi-reference electrode, and also to be able to replicate your work in a way that's reliable. So one way to calibrate and test your quasi-reference electrode is to essentially set up some beaker scale electrochemical measurements that mimic the conditions of your microchip reactor. So for example, use the same electrolyte that you're using in your microchip reactor in a beaker, use the same working electrode, use the same counter electrode, and use a wire of the metal that is your quasi-reference electrode for your in situ measurements, and just run your experiments and see if the potential of that reference electrode drifts over time. If it doesn't, then you're in good shape. You can say, okay, looks like our reference electrode is stable, but definitely take into account that there are geometric differences between the reference electrode that you're using in your beaker scale electrochemistry to that of the microchip reactor. The other thing you do is when you set up this beaker scale chemistry is you can also put in a conventional reference electrode and just measure the potential difference between your quasi-reference electrode metal and the conventional reference electrode. And then all of a sudden you've got a calibration value that you can use when converting your electrode potential measurements. So I have a question in the chat. Will the size of the reference electrode material affect the potential, since it depends merely on the redox reaction? So in principle, the size of the reference electrode should not dramatically impact the potential, because it's dictated simply by the redox processes or the redox couple that's formed on the surface. Another way to calibrate a quasi-reference electrode is by an internal calibration. And you can use something like a ferrocene containing electrolyte or a ferrocyanide containing electrolyte. And those two couples that I mentioned are essentially simple and reversible one-electron redox pairs. And when I say reversible, it means they don't have high overpotential when they switch between the Fe2+ and the Fe3+ state. So if you're running, so you can insert your ferrocene containing electrolyte into your microchip reactor, run CVs along the lines of what Rod Smith was showing in his workshop a couple of months ago. And essentially what you can do is when you look at that CVs, you can see exactly where that ferrocene redox potential is. That's a well-defined value that you can also use to calibrate your quasi-reference electrodes. You can say, okay, I know my gold quasi-reference electrode is +3 volts versus ferrocene. And then all of a sudden you've calibrated your reference electrode to a well-defined value. Then, okay, I'm pretty much out of time. I was going to show some results of in situ TEM, but essentially what we're able to do is we're able to track crystal structure or phase of materials as a function of potential, as well as the structure of these materials as a function of potential. Ahmed Abdallah, a PhD student in my group, did these measurements. Initially did these measurements versus the platinum quasi-reference electrode he was using. But as a good electrochemist, he did his calibration. He switched them all to RHE scales, and we're able to show the phase structure of these materials as well as the morphology of these materials as a function of potential. Just some take-home messages. Make sure you're being careful with your reference electrodes. Make sure you're taking care of them, you're calibrating them properly. And then opportunities for development. I think improving our understanding of quasi-reference electrodes and establishing best practices for their use is important. as well as developing reliable quasi-reference electrodes that have the stability and the convenience of a conventional reference electrode, for example. With that, I'll just highlight McMaster is home to the Canadian Centre for Electron Microscopy. If you have any electron microscopy needs, we are here for you. Facility manager's name is down there. You can email me if you want to get in touch with him as well. Here's an overview of some of the areas that our research uses. And most importantly, I would like to thank my research team. We're growing steadily. I started McMaster in 2019, and we're already at about 12, 13 members, and we have several collaborators and funding agencies that I would like to thank. Last but not least, I'd like to thank all of you. There is my email, there's our website. Feel free to reach out if you have any questions, comments, thoughts. Always happy to chat. Thank you very much.
Awesome. Thank you so much, Drew. This was fantastic. I think everybody learned a lot. If anyone has any questions, feel free to post them in the chat. I have a couple too, so I can maybe start. So I know, like, I work a little bit with like lithium metal batteries, and I know people, for example, in these sort of organic systems often use lithium metal, for example, as a reference electrode, even though it doesn't work very well as a quasi-reference electrode. And there's been some like recent recommendations to switch to something like lithium iron phosphate. I was just wondering if there's a way to kind of predict what materials would behave well as a reference electrode if you're working in a system where you can't really use conventional materials and you have to use these quasi-reference electrodes, or if you kind of have to just do trial and error and see what works.
Yeah, so that's a really good question. And first off, I'll apologize for running a little long on this. I aim to give 10 to 15 minutes for questions, and I just babbled on. But
all good.
Long story short, what I generally have done in the past is use a little bit of chemical intuition, like look up how different materials perform or behave in different electrolytes and with the different species they'll come into contact with. And so down select. And then, yeah, it comes down to trial and error as well. Like, it's impossible to predict everything with chemical intuition. And that's also why I encourage students, I encourage researchers to write these things in their papers, write their thinking process in papers, come up with protocols used for the selection, the use, the validation of quasi-reference electrodes, so that students like you who kind of wanted, you know, your research is not focused on developing a quasi-reference electrode, it's focused on some other practical problem, I'm guessing, can allow you to kind of take that knowledge, take those tools, and not spend, you know, months of your life reinventing the wheel. So I don't really have a perfect recipe to answer your question.
Yeah.
But chemical intuition, reading the literature, and then trial and error is kind of the best that one can do.
And if you want to validate those then, and if it's like an organic system or something, how would you validate it against something like silver chloride or something if you're using organic solvents in like a totally different system?
Yeah, that's a tough one. I don't want to speak. So I have not really done much organic electrochemistry, and I know that is a big area where pseudo or quasi-reference electrodes are used. Probably I would think about using those ferrocyanide redox couples and see how your reference electrode made your lithium electrode drift versus time. But that's like very scratch the surface kind of thinking. True. For me. Yeah.
Yeah. No, I'm just wondering because I definitely don't see a lot of that in papers either. So it's good to know if there's any good ways. Probably have to dig in a little bit to find ways to make sure everything's actually calibrated.
And to be honest, most of the literature out there is on organic, you know, non-aqueous electrolytes. And that's a big reason that WatECS did not include that in the paper, because there is so much in that space, whereas it's kind of a somewhat new area in aqueous electrochemistry. So that's why we kind of focus on that a little bit, as well as that's more relevant to the research that we're doing in our group. But there is a lot out there. How good it is, I can't comment.
No, that makes sense. That's fair. I don't know if anyone else has anything else they want to share in the chat. I guess other than we had the previous questions about the sort of copper silver system.
Yeah, I can go back to that and actually A big question. We've got a big
Oh,
question.
okay. Keith.
Okay.
Feel free to also come off mute if you
Yeah,
want
I think
Keith to explain.
this is like a
Yeah.
back and forth type question. Yeah. Keith,
Yeah, I figured this one might need some explanation and background. But yeah, essentially, I've got a cell where I'm measuring like potentials across ion exchange membranes. So I don't really have like working or counter electrodes. Like I do, but they're like external to the cell that I'm measuring.
Mhm.
And I've had to use these platinum wires as probes because I can't fit any reference electrodes into my system.
Mhm.
And I've got like a repeating cell unit where there's two different membranes and then two different flow compartments.
Okay.
So across the full cell, like the two flow compartments have different pH, but I have like repeating units. So I guess I know that there's issues with platinum wire. Like if you look at a Port-Bay diagram, platinum will have very different electrode potentials at different pH.
Mhm.
So I guess I have two questions. One is like, if I want to measure a full cell voltage, I can just stick it in two solutions at the same pH across a full cell. And as long as they're identical conditions, maybe I'll start with the other question. If I'm sticking to, like, if I want to measure the potential across the membrane, one is like a high pH, one is a low pH. Is there an easy way to correct for that? Because it's basically just measuring the potential across these two platinum wires on either side of a membrane.
Yeah. Okay. And it sounds like you're using like a bipolar membrane. Is that?
Yeah, that's right.
Yeah. Okay. Yeah, that's okay. That's a tough one. I'd have to think about that a little bit. My gut instinct is just do some sort of external calibration. Is the pH of your solutions changing?
No, I mean, I guess in operation, like once it reaches a steady state. No.
Okay, and do you know what it is at that steady state?
Yes.
Okay,
Yeah.
so what you could do then is do kind of that beaker electrochemistry I mentioned. Just make 100 mils of whatever that electrolyte solution is at that defined steady state potential. Butane torch your platinum wire, put that platinum wire in there, and then stick a silver chloride or an RHE or whatever in there and just measure the potential difference. And then you can calibrate it that way. So you'll know, okay, my platinum wire is at 0.7 volts versus RHE.
Okay. And then do it at each condition, and then I can use that as my way to calibrate it.
Yeah, okay. And then if you want to eliminate the impact of pH or correct for that, then you could go to thereafter convert to SHE, standard hydrogen electrode, which is pH 0 or 1.
Yeah, based on those calibrations.
Yeah, that might work. I can't think of anything. I might be missing something, but I can't think of anything that would be a roadblock to that approach.
Yeah, that makes sense. And then I guess the other question is like, so like I've got like repeating cell units. So like I've got two streams at low pH. And if I just have the two platinum wire probes across like in those same stream, I should be able to get the cell potential without any calibration issues because it's identical conditions. Is that right?
Okay, yeah. I think so. Yeah. Because you're just measuring the potential difference between the two, and then that's your delta V, right?
That's right.
Yeah, I think so. I think that sounds right. Yeah.
Okay, that's good. And then I just had another quick question, just because I know you mentioned somewhere in the chat or in your talk that platinum wires have issues with chloride. Is that, or like chloride contamination? Is that, because I do have chloride ions in my system?
Yeah, I think that was a bad. That was a bad example in your context. Platinum can have problems with chloride ions when you go to you increase the potential significantly. You're probably not doing CVs or anything like that with your platinum wires, so I don't think it'll impact you.
No, yeah, it's like 1 to 2 volts kind of thing.
Okay, yeah. As long as you're not doing like crazy CVs or anything, you should be fine.
Okay, that's good to know. That, yeah, that worried me a little bit. Okay, thank you so much. That makes a lot of sense.
Yeah, of course. Platinum is generally, if you've got any kind of like small organics or carbon-based species, platinum gets plugged up pretty easily with those. But other than that, it's pretty good.
Okay, awesome. Thanks so much. And that was a great talk, by the way. You know, great to hear from you. So thank you. I learned a lot.
Awesome. Good. Yeah, I hope it helps you. And you can feel free to reach out to me in the future if you have questions. I'm also kind of curious now if that approach works. So if it does, let me know in a couple months when you test it out.
Yeah, because all I can see is like wildly different potentials, and I know it's wrong. So, but yeah, that's a good idea. I'll try that out and let you know. Thank you.
Yeah, awesome. Yeah, good luck.
Thanks.
So I think there's a few more now in the chat. One from Moyne.
Okay. I'll start with Moen. How do we go about designing and building a micro electrochemical cell? Are there commercial micro electrochemical cells that can be used for in operando experiments? So, so yes, there are. So for example, those transmission electron microscopy measurements I was showing with the microchip reactor and all of that affixed into a transmission electron microscopy holder, all of that is commercially available. On another hand, for the sticks and measurements that Chun Yang and we're doing with Professor Adam Hitchcock, you can buy these microchips. You can even custom design them. There's a bunch of companies that make them. But sometimes your balance of plant, like how you get the electrolyte in, how you connect all the pins to your potentiostat, sometimes you need to kind of custom rig that up. And there's different ways to do that, like building microfluidics or 3D printing cells. So in some instances, yes, there are commercially available ones. In other instances, depending on what you're trying to do, you might have to design your own. Okay, and then Anne asked, doing research in supercapacitors, great. Okay, so the question is, you're doing experiments, doing supercapacitor measurements in a 3-electrode cell versus a reference electrode. But many literature report a 2-electrode cell. I'm guessing in the literature you're seeing where they're reporting a 2-electrode cell is they're essentially building a supercapacitor device, either a symmetric supercapacitor device or an asymmetric supercapacitor device. But then some would report 2 volts of voltage. So if you're talking about the 2 volts of voltage, it sounds like that is just the voltage difference between your anode, your cathode, your positive and your negative electrode. Seems a little bit high if it's aqueous. If it's non-aqueous, that would work. But
sorry, yeah. I was asking about if some people report -2 volt.
Oh, I got it. Okay, I got it. So yeah, I think it's just, so in the electrochemical device, negative, it all depends what they're defining as their positive and their negative electrode. So I wouldn't worry too much about that. I would just look at it as 2 volts. I think it's hard to say when I haven't seen the paper, but I think they're probably just, E cell equals E cathode minus E anode. They're defining one as their cathode, one as their anode, and reporting that value. That's my thinking. Based on what you're interpreting from that paper, and does that make sense, or is there something we're missing here?
So some people in literature, they will report, let's say, oh, my supercapacitor works in the voltage range from -2 to 2 volts, and that's what confusing me. Let's say I chose something to be the cathode and the other thing is the anode. -2 volt versus the cathode and +2 volt versus the cathode is something very confusing to me.
Yeah, because so if you're working with an anode and a cathode, those potentials are essentially floating. So, if you apply plus 2 volts to your device, you could, let's say you have a reference like electrode in there. One of those electrodes could be at, plus 1 volts versus RHE. The other one could be at minus 1 volt versus RHE. Then you just flip that. And then one is at minus 1 volt versus RHE. The other one's at plus 1 volt versus RHE. I think that's where the confusion might be coming. Like the counter electrode is also the reference electrode. Yes, but I wouldn't look at it as such. I would look at it as one is the anode and one is the cathode, or one is the positive electrode and one is the negative electrode. When they go from minus 2 volts to plus 2 volts, they're essentially just flipping the potential of each of them.
Oh, that makes a lot of sense. Thank you so much.
Yeah, hopefully that helps.
Awesome. I think that's most. Oh, no, we have one more. Which quasi-reference electrode is ideal for studying in situ CO2 reduction, in your opinion?
That's a good one. That's a good one. So one issue with platinum is hydrogen. Platinum loves to form hydrogen redox pairs. In other issues, carbon-based organics, platinum likes to get plugged up by carbon-based organics. Gold could be a good one because it does not really bind with carbon-based species. It doesn't form strong redox couples with hydrogen. Gold could be good, but I've never done a systematic study and never really carefully probed that, because we're just working with what is available from the commercial manufacturers at this point in time. But that's a really good question. If I had to guess, once again, going back to this chemical intuition, I think gold would be pretty good, but I'm not sure.
Awesome. I think that's all of the questions, unless anyone sends any last minute ones. So I just want to thank you again, Drew, for today's workshop. It was super informative. I think everyone learned a lot, and also learned a lot of the sort of best practices and practical aspects of using these. So thank you so much again for presenting, and thank you to everybody for attending. I think many people asked about recordings of this. So we have been recording this workshop. It will be posted on our website. We can also send it out to the attendees. If you registered for this event, we'll send you the link. So if you want to revisit it at any point, we'll have that posted hopefully in a couple of days. So yeah, thank you all. Thanks for attending. Thank you again, Drew. And yeah, have a great rest of your day, everybody.
Thanks, Jillian. Thank you all so much.